Williams syndrome and SVAS are relatively rare conditions. There's been a reported prevalence of Williams syndrome in about 1 in every 75,000 individuals. And the prevalence of non-syndromic SVAS, that is when we just have SVAS and not this genotype of multiple genes being deleted, we don't know what that prevalence is in SVAS. Presbylaryngitis, however, is very common. Studies have shown that approximately 29% of individuals over the age of 65 at least exhibit symptoms and signs of presbylaryngitis and experience voice problems as a result. That corresponds to over 10 million individuals in the United States of America, at least reference to the last census figures. In both Williams syndrome and SVAS and presbylaryngitis, a common thread is this hypothesized link between altered or disorganized elastic fibers within the vocal fold tissue. However, we haven't been able to really test that theory by actually characterizing the vocal fold quantity or vocal fold structure within vocal folds and relating that to acoustic properties of the voice. Because we can't go in after recording somebody speaking and grab vocal fold tissue and put it under a microscope. So we've been somewhat limited to this point in making that direct structural functional connection. The human vocal folds are attached to cartilages in the larynx. And the vocal folds themselves are constructed of muscle, epithelium, and extracellular matrix. And most of this extracellular matrix lies between the epithelium and the muscle that's deep. The vocal fold extracellular matrix is composed of a lot of different types of proteins, but one of these that is abundant in is elastin. And elastin is the primary protein constituent of elastic fibers. And one theory that's been attributed to vocal fold elastic fibers is that they allow the vocal folds to displace and then return to midline during the process of phonation or voice production. And in doing so, it's been reported that their normal function is crucial for normal voice quality and normal vocal abilities. That is, elastic fibers need to be organized correctly, in appropriate amount in the vocal folds, and distributed in the right regions in the vocal folds for your vocal folds to vibrate normally and produce a normal sound. Most of the vocal fold elastic fibers are located in the middle of the vocal fold in a region called the intermediate lamina propria. The vocal fold muscle is deep to the elastic fibers here, and the epithelium is superior. And also surrounding elastic fibers are regions of collagen, both superficial and deep to the elastin. So the elastin is in green here, and I don't know if you can see that well or not, but where I'm going here. Okay, so this is where most of the elastic fibers within the vocal folds are located. Together, collagen and the collagen and elastin layers of the lamina propria have been termed the vocal ligament. And we know that vocal fold lesions which disrupt this vocal ligament in some way, for example, vocal nodules, result in significant voice irregularities, significant problems within for voice production in an individual. So it's possible that changes to the elastic fibers in the vocal folds can alter voice quality. Evidence comes from aging, from genetic disorders, and other vocal fold lesions affecting the regions of the vocal fold. But as I've already said to date, this is just theory, because we haven't been able to directly go in and get structure and relate it to the actual function, which would be the acoustic properties of the voice. So this background led to a need for my line of research and also the specific project that I'm going to be talking to you about. We know that human genetic diseases affect the elastin gene and have been anecdotally linked to voice abnormalities. We also know that vocal aging affects voice quality, and it has been suggested that that results from altered vocal fold elastic fibers. However, there's a significant lack of evidence which demonstrates how actual alterations in elastic fibers or the distribution of elastic fibers within the vocal folds influence those vocal abilities. So studies that investigate this problem to find this answer are needed, and they are critical to furthering our understanding of how genes which regulate vocal fold structure and vocal fold development, are altered and influence things such as things such as aging, things such as health, and disease states. So it's also possible, I believe, that increased knowledge of the genotype-phenotype relationship should lead to advances in treatment of vocal fold structural abnormalities that are related to elastin fiber issues. So NIDCD has specifically called for studies investigating genetic influences in the development of communication disorders and for the use of animal models or animal model methodologies to address those issues. The series of studies I'm going to present, in my opinion, present a significant step in addressing the lack of understanding currently in how the elastin gene and the elastic fibers that the elastin gene is responsible for getting produced influence voice production. So a long-term goal of the project I'm going to be talking about is to direct and focus additional methodologies for a larger program of research, which I would need a larger amount of money to pull off. So the current proposed project is really the latest generation of a programmatic line of research that I've been conducting for a number of years. The initial and long-term focus of this research is investigation of human vocal function produced by actual humans. However, as I'll show, the line of research as it is now requires an animal model for further investigation. Now, mouse models have been developed to study the cardiovascular system, which share the exact same genotype of humans with SVAS. That is, they were developed to study SVAS in an animal model. Additionally, mice have been used to study laryngeal aging, specifically as it applies to what happens to collagen fibers within the vocal folds. So the mouse has already been demonstrated as a viable translational model for studying vocal fold structure, and mice with the exact genotype that I would be interested in studying have already been developed. So the initial studies in this line of research focused on voice quality in humans with these elastin gene abnormalities, SVAS and Williams syndrome. And our data demonstrated that these individuals exhibit voice quality characteristics which are significantly different from normals. These differences were both perceptual, that is, listeners could hear the difference in the voice, and they were able to be measured acoustically with instrumentation. So both subjective and objective measures of voice quality and vocal function were significantly different in individuals with these elastin gene abnormalities compared to individuals who were normal. Some of the specifics related to voice production were that individuals with this elastin gene abnormality had an overall lower fundamental frequency of their voice. The pitch was very low. They had a reduced frequency range. They couldn't produce the wide range of frequencies that normal individuals can. They had both a breathy and a hoarse voice quality. And overall, voice abnormality was more severe. It's more severely affected than in normal individuals. So I wasn't able to go in and harvest vocal fold tissue from those humans that I recorded, but I am able to harvest vocal fold tissue from mice. And this is exactly what the mouse model is for in the context of what I'm using. So when I was first looking into utilizing a mouse model, I looked for studies which demonstrated that elastic fibers are actually present in those vocal folds and showed me where in the vocal folds the elastic fibers were. But those studies didn't exist. So I had to actually develop the mouse model for studying elastic fibers within that vocal fold tissue. So I took normal mice. These were wild-type mice. They have no altered genetic code in any way. Got some laryngeal tissue and stained for elastin. And the data showed that there definitely is elastin in the lamina propria of the mice. And just as in humans, it's abundant in the vocal fold tissue of the mice and in the same regions as it is in the humans. And other research which looked at collagen within vocal folds of mice found really the same things, that just as in humans, it's in there, it's in the same regions, and we would theorize that it would function in the same way as in humans. And so in this slide here, this is epithelium, this of course is muscle, and these dark strands in the middle are elastic fibers. If you threw up a human vocal fold, the layering is a little bit different, but if we just stain for elastic fibers, it will look exactly like that. Okay?